A warm welcome to one and all. Myself, Dhanashree Mai, on behalf of Gokul Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, welcome you all to this guest lecture series. Practical approach with theoretical knowledge is the need of hour, and guest lecture will open a window in which our students and we itself as a faculty are enriched with knowledge and latest updates on a particular field by an experienced people. So, for enriching ourselves, our knowledge about pharmaceutical innovation, we have Dr. Pavan Sancheti, Senior Principal Scientist, Glenmark Research Center, Navi Mumbai, with us today. Sir has excellent throughout academic. Uh, Sir has completed PhD in Pharmaceutical Science from North Maharashtra University in 2017, MS in Pharmacy from Naipur Mohali in 2000, and B Pharmacy from SPPU University in 1999. Dr. Sancheti has a huge experience of 19 years in drug discovery and development with special focus on non-clinical safety and toxicology. He has worked with leading research-based pharmaceutical organization in India, in India like Dr. Reddy, Ranbaxy, Advinis Therapeutics, and Glenmark. Sir also has experience of providing non-clinical toxicological strategy for multiple small molecules, that is, novel chemical entities and generics and specialty, as well as the large molecule projects like novel biological entities and biosimilars, right from early discovery to the late clinical development. Sir has developed almost most of the novel drug candidate as an achievement. As a part of team, he has successfully discovered multiple novel drug candidate, small as well as large molecule, that is chemical as well as biological molecule. Sir is also working on risk assessment for pharmaceuticals and calculation of OEL and PDB, that is permittal daily exposure and occupational exposure limit for safety purpose. Sir is also dealing with national and international regulatory requirements for drug development. Sir is also a member of Social Society of Toxicology USA. With this short introduction, I welcome you, Dr. Sancheti sir. Welcome, sir. Sir,
Yeah.
today we are, dear students, sir is going to talk today on innovation of pharmaceutical industry, the process of drug discovery and development. Now, I sharing the screen with you, sir. Please start your session.
All right. Thank you so much. Thank you so much for a nice introduction about my profile and my experience. And it's really my pleasure to, have this opportunity to share my experiences and learnings that I have got over a period of 19 years of my career spanning multiple pharmaceutical industries in India. So, giving any talk or, guiding the students or sharing my knowledge with the college students is really something that I want to do and I like because I can really connect with the students. I look back 20 years back when I was a student. I can connect. What were the doubts, what were the questions I was going through, or some of my friends were going through. Because the college when we get into the B pharmacy, the problem with student is first year. Uh, Students spend understanding what is pharmacognosy, pharmacology, understanding and getting familiar with the terminologies and the college. Second year is little bit of okay, and then third year onwards again the pressure starts mounting, and the final year becomes very very tough for students because they are at a juncture where they need to make a decision whether they want to go for MBA, they want to go for masters, what do they do in the career, they want to opt for academics or what is it. And that's where a lot of guidance, a lot of understanding, if it is there, I think it would just help the students understand what path they want to really opt for, how they want to move forward in their career, and that's why I know I really enjoy sharing my experiences and give some kind of a guidance to the students so that even if it helps to them in a smaller way, I think that's a great thing, great achievement. So with this introduction, I begin my talk today. So today I'm going to talk on innovation in pharmaceutical industry, particularly I'm going to focus on the process of drug discovery and development. Okay, and if I had to just summarize it in one sentence, what I'm going to talk, it's about the journey of a drug, the drug from the laboratory conceptualization of an idea, laboratory experiments to reaching that drug to the bedside for the patient in the clinic or in the hospital. And that's the entire journey of the drug. And, it appears very simple, but I think it's pretty complex. And I want to give you a very high level understanding and some kind of a flavor for what this journey is for any drug, from laboratory till it reaches a patient. All right. So with this, I begin my presentation. This was one slide I had kept just to tell you guys my journey so far, but I think my introduction has already been done. I wouldn't spend too much time, but just to brief, from 1993 onwards, I am associated with the pharmaceutical field, and I did my diploma and then B Pharmacy from Indian VPS College, then NIPER, and then PhD while working with Glenmark. So, and my industry experience has been like 2001 till date. I'm working purely in the drug discovery area. So I will not spend too much time here, and let's get started with our topic. Before we begin, and I start talking on my slides, I think the most important aspect is let's keep it interactive, because this is a virtual talk, and it's very difficult to engage all the people. So I don't know the body language, I cannot see the people. So I think it's most important that we stay interactive. Ask me any questions, any language, Marathi, Hindi, English, whatever you're comfortable with. Anytime during my presentation, end of the presentation, I'm perfectly fine. But let's keep it interactive. Uh, my slides are not very heavy. I have kept it very light. I want to have more interaction, more questions, and so that I can share more of my experiences. So with this, let's begin with this topic. So cycle of innovation and research. Uh, we always hear innovation. We always hear research. What is the difference? What is innovation? What is research? And Dr. Nicholson said it very rightly that research. Uh, Give me a second. I just make my pointer so that I can highlight. All right. So research and development is the transformation of money into the knowledge. So you have money, you do research and development, you do all those efforts, and you develop some knowledge, some database, some knowledge base, right? And then you use that knowledge for innovation, and that innovation using that knowledge will help you again transform this knowledge into the money. So this is a continuous cycle of innovation and research and development. So innovation helps you generate money, and you use that money to again do R&D and invest further and generate more knowledge. So just wanted to give you this flavor of what innovation and research is, and then we can get to the next slide. Okay, so diversity of pharmaceuticals, right? So we all are I mean, my major target audience today is, I think, all pharmacy students, and all of us deal with the pharmacy subject. We take medicines. Our relatives, our parents take medicines, but unfortunately, I don't know how the situation is today. Maybe the syllabus has changed, but I remember the syllabus when I was a student. It's mainly focused. The complete studies, the entire curriculum and syllabus is focused on chemical entities or the small molecules, like all chemical structures like aspirin, verapamil, atenolol, all kinds of drugs, that we check. But I think there has been a major shift in the new drugs. And from a small chemical molecule, there is a major shift that's happening globally to, peptides, some kind of growth hormones, and most popular one as on date is monoclonal antibodies. These monoclonal antibodies are very, very hot, and they are really the I would say hot cake for research all over the world as on date. So I hope slowly our curriculum and syllabus for the students would also change, and they will also start learning about these antibodies and a lot of are biological molecules, peptides, hormones, growth hormones, that kind of stuff. So if I have to give you a little more clarity on what these drugs are, so look on the left side, this is the small chemical molecules, and this is what we know always. As a student, we always talk about these drug molecules, we study these drug molecules, but there are a lot of other categories, and all these can be clubbed as a biologicals or a large molecules. You have peptides, you got proteins for therapeutic purpose, you got enzymes, a lot of enzymes are used as a therapies, interferons, a lot of cytokines are being used for treatment. Those who are right now familiar with the COVID treatment, one of the major treatment for COVID is an antibody drug called tocilizumab. So that's an anti-IL-6 antibody. So that's a biological molecule. That's an antibody. So cytokines, you have hormones. This is the monoclonal antibody. There is also a category called antibody drug conjugate, wherein you have a monoclonal antibody and you also have a anti-cancer cytotoxic chemical drug. You combine these two antibody and a small molecule, and then that makes it a drug antibody drug conjugate. And the last one and the latest one is a gene therapy. I don't want to get into the details for what gene therapy is, but just to give you a flavor to what level the science has advanced, to what level the research is happening all over the world. Gene therapy is mainly for cancer, wherein what we are doing is we take out if there is a particular patient who has a cancer, we take out the cells from that patient, do some genetic transformation in vitro in the laboratory, expand those cells, grow those cells, and inject those cells back into the patient. And that gives a phenomenal result. That gives a wonderful therapy for the cancer. This is the latest advancement in science. So the message I'm trying to convey here is small molecules is, of course, the major part of the treatment. These are the major drugs. And historically, we have been working on them. And I think as on date also, many companies are working on small molecules. But there is a shift that is happening. There is a change in the thought process. There is a change in the requirement globally. And the science is advancing. And, there is a more research happening in this biological areas and getting all the way to the cell-based therapy, gene therapy, where you have a customized therapy. You're taking out the patient cells and giving it back to the patient with the genetic modification. So this is very interesting area that's coming up. All right. So having said this, now let's. So this I don't want to complicate too much, but I wanted to give you some flavor of where the science is and where the globally the research is moving. And now we'll focus on our topic. So when we talk, this slide is all about the R&D. So as a student, I remember I'm sure the students today in the college are much more familiar and much more aware, and they might know this. But uh, we always knew when we say research, what you want to do after B Farm or M Farm. And if anyone says I want to go to the pharmaceutical industry in the research, I think most of the people think about formulation development as a research. Again, that's not bad. That's good. I mean, that's where the Indian industry is flourishing. Most of the Indian companies are generic companies, and they are doing formulation development. But that's one part of an R&D. And essentially, when you talk about formulation development, you are not discovering a new medicine. You are not coming up with a new molecule. What you're doing is some bigger company like Pfizer or Merck or Glaxo, they have discovered a medicine 15 years back. That goes off patent, the patent gets expired, and you are trying to launch the same medicine with doing some formulation development. That's formulation development. But what I'm going to talk is about the new drug discovery. That's coming up with completely new molecule, completely innovative molecule. So if you look at this picture, you see on the x-axis, you see complexity of the research, the amount of money that you need, investment, the time that you need, and the risk that you are taking for the research. That keeps on increasing on the right side. And on the left side, you see on y-axis, you see the returns on the investment. They also keep on increasing. So that's the basic fundamental of life, that you have more you invest and more risk you are ready to take, more are the returns. So if you see this value chain for the pharmaceutical research, start with generic. This is the basic generic. The patent gets expired and you launch that generic molecule. Generic plus is little bit complex projects. People do more complex molecules where intravenous IV formulations, etc. There are some super generics or complex generics wherein you combine two different molecules and then try to come up with a combination. But all these are four categories are for the small molecule drugs, that is the chemical drugs, and they are some or the other way they are generic drugs. You are not bringing the new drug to the market. All you are doing is existing drug you are combining with something else, or you are changing the formulation and you are bringing to the market. So this is a low cost intensive. You don't need to spend a lot of time and money, but the returns are also low. Little bit newer area is biosimilars, like how you have generics for the small molecules. For large molecules and proteins, you have biosimilars. And this is a completely separate topic, which I can talk about some other time. But this is a little more complex than generics. But again, here as well, you are not discovering a new medicine. This is still the same medicine. You are trying to copy it and launch in the market. And then this is my favorite topic, and this is where I'm going to focus today. That's NCE, or the novel chemical entity. I mean, we historically call it novel chemical entity because novel small molecule, but there are also large molecules that are happening right now. As I explained in the previous slide, you have proteins and monoclonal antibodies and biologics. So they are called novel large molecules, and chemical drugs are called novel small molecules. So let's understand the difference. Small molecules are chemical drugs, large molecules are biological drugs like antibodies and hormones, etc. So this NCE is actually a completely novel research. You see in the value chain, it is at the top. You need a lot of very complex research, lot of investment, lot of time, lot of risk. But you look at the returns, it is at the top. The returns are very, very high. So with this premise, let's get into how complex is drug discovery and development is. Pretty simple, right? This slide shows it's simple. I'm joking. It's not really simple. It's pretty complex. Drug discovery process is very, very complex. And this slide is not to complicate it further for you. I just wanted to give you a little flavor of how complex is this entire process and how many different functions and departments need to come together and work for a project that lasts for 10 to 15 years. It's such a long time, and you need to really stay focused and work as a team, so many different functions. I don't want to go through the details for this slide because it's very complex. I will simplify it in my next slide. So if you look at this funnel, this is a very popular term called drug discovery funnel. So what do you mean? When you drug discovery means you are trying to come up with a completely new molecule. So you start with roughly 20 to 30,000 drug candidates. In the chemistry lab, you synthesize a lot of molecules, right? There are a lot of chemists, organic chemists, medicinal chemists who will be working on this. They make a lot of molecules, and the ratio is, this is a step one. Then as you go down, this is a funnel, right? The number of molecules keep on going down. So from 20,000 to 30,000, you when you go to step two, where in step two you look at step one, you look at discovery screening. Like you are looking at these chemicals, whether they are active or not, whether they have potency, whether they are binding to the target or not. And a lot of these will get filtered out. And from say 20 to 30,000, you may come down to example 10,000. So step two. Then step two, you do more extensive screening. The 10,000 might become 5,000 or 3,000. Example I'm just giving. Go to step three, you do a lot of drug metabolism and pharmacokinetic screening that will further narrow it down. And this process continues. You start with 20, 30,000, and by the time you go to step five, wherein you approach regulatory authorities for getting their approval, to launch it in the market. It's only one drug that makes it to the market. Just imagine the quantum of work that you're doing, starting with 20,000, 30,000 molecules, and then it results into only one successful drug candidate that goes into the market. Okay, and that's where the biggest challenge is. And how much time does this take? You look at this side, 10 to 15 years. Okay, and you do this research for 10 to 15 years, and there is no guarantee that after 15 years this molecule will be successful and it will go to the market. It may fail at any of these stages. It may fail in step two, step three, step four, step five. We don't know. It might even many a times the drug gets launched into the market and then it starts showing some side effects in the patients, and then the drug gets withdrawn from the market. So the success rate is very, very low. And how much does it cost? $1 billion to $2 billion American. And just to simplify it further, $1 billion American is 7,500 crores. Okay, it's a huge, huge amount. So discovering a new drug and bringing it to the market takes roughly 7,500 to 15,000 crores, depending on a lot of other factors. That's such a hugely expensive affair and such a time-consuming affair. So whether it makes sense to go for this research, I will come back to this in my subsequent slides towards the end of my presentation. But before we go there, let's understand little more details about this drug discovery process. So I have set the context on what this research is and how complex it is, how much time it takes. Now let's understand the actual process. If you look at this slide, these are stages of drug discovery. This is a very simplified slide and it cannot be simpler than this. It's very complex process. But to make it simple, you start with identifying a target. Okay, so this process has discovery stage and a development stage. Discovery stage is where you are identifying a target. So what do you mean by target? I will explain that in my next slide. But it's precisely the receptor or the enzyme that you want to hit. That's a target. You need to identify that. This is the most important aspect. If you go wrong on this aspect, your entire process down the line will go wrong. So you need to be very correct and selective in what you choose as a target. Then your chemistry efforts start. You put a lot of chemistry efforts. You synthesize chemical compounds and see whether these drugs, I would not call them drugs, I would call them the chemicals, whether any of them is binding to the target and showing some activity. Okay, you establish activity. You say out of 2000 compounds, you get 500 which are showing good activity. Okay, what you need to do is, it's not just one assay, you do multiple experiments to optimize further. From say 1000, you come down to 500, 500 to 100, 100 to 50. You need to keep on bringing it down, and you keep on running more assays, more complex assays, more experiments, and do some pharmacology experiments in animals to see whether whatever you are seeing in vitro, whether that's translating into in vivo situation, and you are able to see some activity in the animal models of disease. Once you get that clarity, then you select a candidate. Okay, I started with 10,000 candidates. Now I'm down to 5 candidates or 10 candidates with lot of funneling approach. Now what do I do next? You select one or two or three or four candidates probably, which look the best among all of those molecules, and you do the safety testing. As a pharmacist, most important thing for us is how efficacious is drug is important, but most important is how safe is the drug. You don't want to cure an infection of a patient or a headache of a patient by giving a drug that's going to cause cancer or that's going to cause asthma or some other liver toxicity. Absolutely not. So it's absolutely important that the drugs that you give to the patient or drugs that you research or bring to the market are very safe. So then the safety testing. And then all this is discovery, and you do some testing in animals, and then you move to humans. Once you are confident about animal data, go to humans. Once it goes to human, there are multiple phases of trial: phase one trial, phase two trial, phase three trial. All these different phases of trial take around 7 to 10 years. And you transition through this, and then you go to FDA or regulatory agency, and that's where you get an approval to launch the drug in the market. And then, as I was saying, there is a phase four trial that's called post marketing surveillance or pharmacovigilance. So you launch the drug in the market, that is not the end of your responsibility. You need to keep on getting the data and understand how the drug is doing in the market, whether the patients who are getting that medicine, they are having some side effects, whether there is any toxicity, any issues. There are a lot of drugs which were blockbuster drugs. They were in the market, but they started causing some toxicity and adverse effects in the clinic, and they were withdrawn from the market. So this is the entire process, and in next slide, few slides, we will talk about each of these activity in brief. So let's begin with the most important aspect: target identification and validation. This is most important, as I said. So you want to find out a target on which you want to start your research work. So what do you mean by target? So therapeutic target, in a very simplified language, it's a protein or a receptor or an enzyme or an ion channel whose, or it could be transporter, whose activity can be modified by an external stimulus for therapeutic benefit. So you external stimulus is your drug molecule. So there is a target. You give your drug molecule, it will bind to the target and elicit a pharmacological action. This is what you want to do. What are say examples? As I said, receptors. There are ion channels, transporter proteins, enzymes. So if you are to name some drugs for receptors, you can have a beta blockers, example, atenolol, those kind of drugs. Enzymes, you have ACE inhibitors, angiotensin converting enzyme inhibitors like enalapril, which are in the market. Ion channel inhibitors like calcium channel blocker, verapamil. Transporters, these are the latest drugs, SGLT2 inhibitors for diabetes like empagliflozin. Again, this is not the master list. There are a lot more targets. I am giving you the terms which you are familiar with based on your syllabus and curriculum. So what do you need to look at when you're talking about a target, when you're trying to select a target? What are the attributes that you look for in a target? Most important is what kind of target you want to work on. Receptor, should it be enzyme, should it be ion channel, what is it? Expression pattern, where is it expressed in the body? Is it expressed on the surface of the cell membrane? Is it inside the cell? Is it in the blood? Is it in the skin, brain, where it is? Expression pattern, most important. Then validated versus non-validated. So what do you mean by validated? Validated target means a target, you know that it works, that there is already a drug which is working on that target, and it is known that this mechanism works. That's called validated target. Like say there is a beta blocker that is in the market, we know that this is a validated target. So if I start another drug for beta blocker, it will be an easy path because we know that it's going to work. But non-validated target is something where you do not know whether that target is going to work, and that's where the we do the research. You take a risk, select a target which is novel, and then you try to see whether that's going to work. Then first in class versus best in class. So competitive intelligence, the most important. If any organization or company is going to spend 7,500 crores or 10,000 crores, they look at what is the competition in the market. If there is a drug in the market already, five, six drugs for a particular disease or a mechanism, you don't want to spend 10,000 crore and again bring another drug to the market. You would look for a opportunity where there is no one in the market and you can get better business. So first in class will be for non-validated target, whereas best in class is validated target. Then modality, whether you want to develop an agonist or you want to develop an antagonist, what do you want to develop? Feasibility and availability of assays. Okay, you select a target, it looks very good, but can you really run the experiments? Is it whether the assays are available, whether animal models are available? You need to look at all those things. What is an indication? That's the most important thing. You have a target, and this particular target can run in two or three different mechanisms, two and three indications. It may work for diabetes, may work for hypertension, may for cardiovascular. But then you need to decide as a company or as a research group which indication, what therapeutic area you want to work. And then most important, intellectual property space. You don't want to do something whether there you are not going to get intellectual property protection. If your IP has to be protected, there should be enough space to do the IP and the research. Then only I think you begin this target. Very superficial coverage of the target selection is a lot more complex than this, but this is how we begin with the target. Okay, you are now zeroed in on a target. You know which target you want to work with. What do you do next? Then your chemistry group comes into picture. And what does chemistry do? Okay, you begin with computer-assisted drug designing. This is called molecular modeling. You must have heard the term, wherein you have a crystal structure or a 3D structure, protein structure of the target that we talked about in the previous slide. You put that structure in the computer system, and then you create a virtual library of chemicals, and you see which of these chemicals is binding to this target properly. And this is not very resource intensive because it is all done in the computer system. This is called computer assisted drug design. So this particular method or technique does not give you the final drug, it gives you a starting point. There are also some new approaches that are come up, like traditional synthesis, you will just do A plus B is equal to AB. But combinatorial chemistry or combinatorial synthesis, wherein there are a lot of options, A1, A2, A3, like AN, and then you start doing different permutation combinations. And instead of making one molecule, you can create 1,000 molecules from by using different permutation combinations. Combinatorial chemistry is another tool. Uh, and then you use these two as a starting point. Most important, do intellectual property review that, okay, you are working on this chemical space, but whether some other company has already blocked this chemical space or not. If not, then okay, you are free to work in this space. If already say Pfizer or Merck has already blocked that space, they have filed a patent, you cannot work on that chemical space. You need to find out another chemical area. You have the starting points, you have IP review done, and then you start generating chemical compounds. You synthesize some, say, hundreds of compounds, screen them through in vitro assays, and see out of those 100 or 200, some of them might start binding to the target. Then they are called hit. You know that, okay, this particular chemical structure is binding to the target. That means there is a good probability of moving forward with this particular chemical structure. Whichever structures are not binding, you eliminate those structures, and whichever are binding, you start working on them and refining it further. Then you do a lot of in vitro assays and ADMET assays. ADMET means ADME, These are DMPK, pharmacokinetic assays, and some toxicology assays. You do all these assays and you call lead generation. So from hits, where you have 100, 200 hits, you narrow down to 10 or 15 leads. These leads are like much better than hits, where you have, if hit is like say 100 micromolar, or sorry, 10 micromolar potency, leads will be like 800, 700, 500 nanomolar. They will come down. You start doing more work on lead generation, and then you do lead optimization. You do some pharmacology assays, in vivo assays, and then you do lead generation, and then sorry, you do in vivo assays, and then you optimize the structure further for the properties, and you probably are left with 5 or 10 molecules at the stage. And once you have those molecules, then you know that it is working, it is binding to the target. You screen it through the pharmacology models, in vivo animal models, and that is where it's called preclinical candidate. Once you know that pharmacology, there is an efficacy, it is active in the animal models, it is changing the disease status. You do safety testing, that's toxicology testing, and once you do that, then you have a clinical candidate. So you have done all this process, and then you have come to the stage where you have a clinical candidate. Clinical candidate is the final molecule that you want to move from discovery to development. When you say development, it is a clinical trial. So clinical trials are very, very expensive, and you don't want any clinical trial subject or volunteer to die because of a drug. So once you are 100% sure on its safety from all this process and efficacy, you declare a clinical candidate, and that goes to further development in the clinical trial. And during doing all these things, you develop structure activity relationship. I'm sure the students know that in medicinal chemistry you must be studying SAR, structure activity relationship. Like one particular group, functional group, is important for activity. Another particular group is important for stability, or something like that. And you modify chlorine to fluorine, and your activity goes down. This is all called structure activity relationship. This process is continuous, right from hit generation till you have a final candidate. You continue, chemist keep on doing the structure activity relationship continuously. So you have a role of medicinal chemist, organic chemist, and a computational chemist for CAT here. So we covered target selection, then we got into the details of medicinal chemistry efforts. Okay, what do you do next? Now you have a chemistry team who has synthesized the compound. What do you do next? In vitro biology. What is in vitro biology? In vitro is not in the animal models, not in the living system. You take out the cells from human, biopsy or surgery, say example tumor cells. You prepare their suspension, you grow those cells in the laboratory, and there are some genetic modifications you can do to make them immortal. Those cells don't die. You can convert them to cell line, or you can use them as a primary culture. And then these cells you grow, and then you run in vitro experiments in laboratory on these cells. If you can derive those cells from human as well as animals, and you need both actually, because early stage you develop it from mouse, understand it, if it is working, then you do it for human. And once you have these cell lines ready, you set up the assays, and you use all these in vitro screening technologies. Like say this is a well plate where drug A, drug B, drug C. You can test 10, 15 compounds at a time in one particular plate. You have 10 chemicals that are synthesized by chemistry. You put them in this plate. This plate has these cell lines, or the cells, primary cells, and these cells express the target that we are working on. So you are able to relate, right? And then you are looking at the endpoints, whether our drug is working in these cells, and our drug is working on the particular target in these cells. And whichever drugs are out of this, say compound A, B, C, D, E, F, whatever you have, whichever are working properly, you will take them forward in the next set of assays. So what all screening you do? You look at just a pure enzyme and you do the assay. It could be a cell-based functional assay like the ones that I shown here. Selectivity assay. This is another important thing. So you want that your drug should be, say example, inhibiting a particular enzyme. Okay, that's fine. But what is the guarantee that your drug is not inhibiting other enzymes? Because if it starts inhibiting other enzymes, there will be a lot of side effects, adverse effects, toxicity, and you don't want that. You want that your drug should be very selective, work on only the target that you are selected. You don't want it to work on some other target. So that's where you do selectivity. We have thousands of receptors and enzymes. We screen our drug molecules against all of them. to check which all mechanisms it is working through. And then the safety screening assays, like, you do some in vitro assays, toxicity assays in cell lines. So all this kind of. There is some newer technology that is right now being used in industry, that's called high throughput screening. I don't want to go into the details, but this is mainly robotic because you're getting thousands of compounds. If you start screening them manually, I think it's going to take a long, long time. So you want to expedite the process. You use high throughput screening where robotic arms are there and machines do all the screening. So this is about in vitro biology. What is next? You need to go to in vivo pharmacology, but before that, understand the pharmacokinetic properties of the drug. So okay, you look for DMPK. What is DMPK? I will cover it very briefly. So you take a drug orally, drug goes to your GIT. Okay, that's called absorption phase, ADME. From once it is absorbed, it gets into the blood circulation, gets circulated into the body, it gets distributed into the body. That's called distribution. You all know, you must have studied in pharmacokinetics. Once it distributed, it also goes to liver and a lot of other places. And there is the metabolism process, phase one and phase two metabolism that happens. That's called metabolism. And from once metabolism is done, the drug gets excreted through kidney, okay, kidney or even the fecal matter, and that's called excretion. These are the basic process: absorption, distribution, metabolism, and excretion, ADME process. And typically, this is the graph that you get, right? You all are familiar. I will not spend too much time on this. This is the graph. This is an absorption phase, and there is also some elimination happening. This is a T max and C max, and then here the elimination phase dominates, where your drug goes down and you get an half life. The As the time progresses, the concentration keeps on increasing initially and then goes down. This is a typical profile. What do you do in DMPK assays? You look at in vitro metabolic stability. Okay, so you have made hundreds of compounds, thousands of compounds. You have screened them in the in vitro assays. You know if they are active, they are binding to the target. Now you cannot put all those molecules in animals to check for their pharmacokinetic properties. It's not possible. It will lead to consumption of thousands of animals, and that's not ethical. What you do is you take out liver and separate microsomes from animals, and those microsomes in vitro in the laboratory in the cell culture plate, you test their metabolism, and 100 compounds you can screen at a time. You know which compounds are better. You check for their CYP inhibition. CYPs are the enzymes, right? Cytochrome P450. You don't want your drug to inhibit those enzymes, or you don't want your drug to induce those enzymes. You want to check all those things early on. You look for plasma protein binding. There are a lot of proteins like albumin, globulin in the plasma. Your drug binds to them. You need to understand that. Tissue distribution. Okay, you want your drug to go to skin and treat the dermatological disease. But the distribution of your drug in the body tells you that your drug is not going to the skin. It is staying only in the other tissues. It's not going to skin. Even if your drug is good, it will not show any pharmacological action because it's not going to the target tissue. It is important that you want your drug to be effective in the skin, it should go to the skin. Or you want some CNS drug for brain, your drug should penetrate the blood-brain barrier and reach brain. So you need to do all these studies, tissue distribution studies. Then you do a complete pharmacokinetic profiling in animals, and then the PK/PD correlation. So you have done the targets, you have a chemistry done, you have in vitro biology activity evaluated, you have the properties of DMPK, ADME. Then you get into the pharmacological assays. That's like pharmacodynamics. I think you all are on top of this. You must be studying it in pharmacology. What pharmacodynamics is, what your drug does to the body, right? So essentially, there are animal models of disease that you develop. How do you develop them? So depending on the indication, if it is diabetes, there are models that you artificially create diabetes in the animal models. This example is for tumor cancer. So you take the cells, whichever cells. If you are trying to target lung cancer, you take the lung cells, cancer cells, give them give those injection to the mouse. There will be a xenograph that will be developed, and then you give your drug to the mouse and check for activity. These are disease models developed in the lab. Nowadays, we are also doing a lot of work on the transgenic or humanized mouse. So, because what happens is this is an animal model. Whether this will translate into humans and it will show similar activity in humans, we do not know. What we do is humanized mouse, transgenics. What you're doing is we take the genes from human and induce those genes into animal. And that's why these animals are called humanized mouse. So it is the mouse model but the target that you're working on or the genes are human genes. So whatever results you get here, they are very likely to translate into humans, and you can be confident that okay, whatever we see here in this model, we'll be able to see the similar level of activity in humans. These are very popular but very expensive right now, but they give you a very accurate data. So what do you want to achieve in in vivo experiment? You take multiple doses and face the dose response relationship. You come up with ED50 value or ED max value. ED max is the maximum efficacy. You need to understand how this result is going to translate into human, and then the PK/PD relationship. What is PK/PD? So you have time versus concentration in PK and dose versus the response. But what you need to do is the actual drug concentration on x-axis versus the pharmacodynamic response. So here is a PK on x-axis and PD on y-axis, pharmacodynamics. So you need to be able to relate as your concentration of drug increases, you are able to see increase in the efficacy or the pharmacological action. That's called PK/PD correlation. You need to do all these things. Okay, now you know your drug is working fine, it's effective, it's binding to the target, active in animal models. What do you do next? As I said earlier, most important aspect, you need to make sure that your drug is safe. And for your drug to be safe, you need to do a lot of testing in toxicology. So look at this graph. This is the graph where it's a efficacious dose, and as the dose increases, you start getting into the toxicology zone where side effects start emerging. And with the side effects, then you don't want to achieve these doses in human. You want to stay only in this range, and this is called therapeutic window. Toxic dose and a safe dose or the efficacious dose. You want to stay in between this. What do you do in toxicology? There are regulatory requirements. There are regulatory agencies like FDA for United States Food and Drug Administration. For entire Europe, there is an agency called European Medicine Agency, EMA. There are a lot of guidelines harmonization happening, ICH. You need to do all toxicology studies in a GLP manner. That's a good laboratory practice. There are regulatory requirements that you need to do XYZ prescribed studies, toxicology studies, and only then any regulatory agency like FDA or European agency or Indian agency will give you a permission to go to clinical trial in human because they don't want to risk human life. Then what do you do in the toxicity? You want to test on target toxicity and off target toxicity. So what is this? On target means, say you want to your target is, say example, beta blockers you want to develop. So whatever toxicity you get because of blocking a beta receptor, that's called on target. And off target is along with blocking a beta receptor, maybe your drug is also blocking something else, and that's not something that you want. That's called off target toxicity. So you need to understand these both the toxicities. It is compulsory, a regulatory requirement that you do toxicology studies in two species. One should be rodent. Rodent can be rat or mouse, and another species should be non-rodent. Non-rodent could be dog or a monkey, and we have to do all these studies. And this is in fact the area of my specialization. You need to understand what organs are getting affected. How will this help you? You do a toxicology study in animals, say dog or rat, and you know that in animals there is an effect on heart, or you know that there is an effect on liver. Then what happens is when the drug goes to the clinical trials in humans, you know that this particular drug candidate can damage your liver or it can damage your heart. And what you do is you start monitoring heart and liver in clinical trial. And that's where there is a possibility of these organs getting damaged. Let's monitor these organs. So in human clinical trial, you will look at heart evaluation repeatedly to check that the humans do not have that toxicity or any side effects. Evaluate reversibility. Everything has a toxicity. It's just the dose that differentiates, right? At the low dose, it is safe. As the dose goes up, it becomes toxic. But even if it is toxic, it is important that whether the toxicity is reversible, whether you are able to reverse the toxicity once you stop the treatment. It's very, very important aspect. You check for that. And most important aspect, you look for the safe dose for human. You do all toxicology studies in animals, rat, mouse, dog, monkey, maybe sometimes rabbit, mini pigs, and then you evaluate all that data and make a decision. Okay, based on all this data, this particular dose should be good enough and safe in humans, and you suggest that starting dose in humans, and then you start the clinical trial. Very briefly, what all kind of studies you do in toxicology? You do in vitro toxicity. Your drug is known to cause liver damage, you will do in vitro liver cell assays. You need to do acute toxicity testing. Acute means you give a single dose, very high dose, single, and then you start seeing toxicity. Repeat dose. Every day you administer the drug to animals for 1 month, 3 months, 6 months, 9 months. Maximum duration is 9 months. You need to do, and then see at the end of 9 months or 6 months, you see what kind of toxicity your drug is causing to the animals. Because if when it goes to patient will be taking that medicine every day for the life. So you need to have a repeat dose toxicity. Safety pharmacology, what kind of effect your drug is having on those different important organs, vital organs like CNS, cardiovascular system, respiratory system. Genotoxicity, very important. Your drug molecule might go into the body and interact with DNA and cause genotoxicity. It may change, it may make genetic modification, and that may lead to cancer. You test all the genotoxicity liabilities early on to ensure that your drug does not interact with DNA. Carcinogenicity, you need to do 2 year study, rat or mouse. You give the drug molecule for 2 years and see whether those animals are developing any cancer. Very, very important. Reproduction and developmental toxicity. What happens? Many drugs that are in the market, they are not allowed to be administered to pregnant ladies. Why? Because some of the drugs can cross the placental barrier and make some damage to fetus. That's why these studies are very important, that whether your drug causes any problem to the fetus if it is given to the pregnant animals or humans. And then phototoxicity or local toxicity, you have a drug which is going to the skin, and when there is a sunlight falling on your skin, there could be a degradation of the drug, and that may lead to phototoxicity. You need to evaluate all that. It's very simplified presentation, but it's very, very complex. Now you are done with your discovery phase. You have done toxicology. You have done everything. I know I'm running out of time. I will quickly summarize. So you have done everything else, and now you are ready. You are confident on your molecule. You want to go to clinical trial. You want to test it, your hypothesis in humans. So what do you do? There are three different phases of clinical trials: phase one, phase two, and phase three. And these are just the generic things. Depending on the drug, it varies. So for phase one, it usually takes say 6 months. Sometimes it takes 1 year, and you take limited number of healthy volunteers. And what you test here is you start giving your drug. You get few answers: whether your drug is safe, what are the side effects. If it is not safe, okay, what are the side effects that your drug is causing, and how does the drug work in the body? Is there something happening in the body with your drug? Very preliminary readout that you do in a phase one trial in the less number of volunteers. And here, I'm sorry, the success rate is around 70% because phase one trials are relatively easy. Phase two trials are a bit complex. You do these trials in the patients. Here it says 10 to 50, but sometimes your patients can go to 200, 300 as well, depending on the size. And your trial can last 3 months to 2 years or maybe 3 years sometimes. And what answers you get in phase two trial? Whether your drug is again safe. Here it was in a healthy volunteers. Now you are looking at the patients. Important point: whether your drug is safe in the patients. How effective is the drug? Whether you are, because these are patients, you can actually see whether your drug is effective in the patients, whether it is treating the disease or it is not. And how much dose should be taken, because it's important to define a dose, what dose you want to give. You get all this information. Very, very important trial, phase two trial. Here success rate is around 33% in general. You are clear here, then you go to the next phase, phase three. This is the largest trial. It lasts for around 3 years, sometimes 4 years. Patients can be sometimes in thousands as well, depending on the design of the study. Uh, and here you again look at the large number of patients, whether your drug is safe, whether it is effective, and is there a real benefit that you're getting in this. So whatever you get in phase two, you kind of validate the same findings again in phase three, but with the large number of patients, one, and multiple geographies. This could be at just one center or two centers. Here you take in phase three trial, you take genetically diverse population. So your drug may work on in the Asian population. You want to take African people. You want to take American. You want to test Europeans, because all these people have different genetic constitution. You want to test it across the globe, different racial population, and you check whether your drug is effective. And then once all these three phases are complete, Of course, you entire data package to regulatory agencies. They will review the data and they will give you permission, and then your new drug becomes available to the patients in the market. And then the phase four, once it is in the market, phase four trial starts, post-marketing surveillance. So this is about clinical development. Then what is the role of regulatory agencies? Regulatory agencies are the agencies which control your everything related to your drugs and pharmaceuticals in the respective country. Every country has got national regulatory body. For example, FDA, United States of America has US FDA, Food and Drug Administration. Europe has got European Medicine Agency. Japan has got PMDA. Our own India has got DCGI, we call it Drug Controller General of India. This is based in Delhi. So we any drug development, any clinical trial, any permission you want in India, you need to approach DCGI office. UK has MHRA. Different countries have their own regulatory bodies. What these regulatory bodies do? They review the data that companies submit and then decide whether they want to give us a permission or not. We first time we approach FDA or regulatory agencies, IND application. IND is when your drug candidate is ready and you want to start the clinical trial. Go to regulatory bodies, get the permission here. And from here, for entire clinical trials, you need to continuously keep on interacting. And then once your data is done, then you get an approval. And this is where the regulatory agencies play an important role. They set up a regulatory agencies. What do they do? They set up a process for approval and data requirements, develop and publish the guidance document on what companies should do. They review the complete data, ensure safety of the patient, most important. They advise the companies. Sometimes, agencies like FDA, very, very good. If you go to them, they will not only find out your faults, they will also guide you on what you should do, which will be better. So it's very, very important aspect. And then they also take regulatory action, sometimes to withdraw the drug from the market. Okay, so this is all about, we talked about medicinal chemistry and then we reached all the way to FDA approval. So I covered the entire process of drug discovery. This slide we have seen earlier. I just want to recap very quickly. We talked about the target selection, right? Then we talked about medicinal chemistry efforts. We talked about in vitro and pharmacological activity, established activity, select the clinical candidates. We talked about DMPK properties. We talked about toxicology, safety testing, and then we talked about the clinical trial, phase one, phase two, phase three, and phase four. Phase four is a pharmacovigilance. We covered all these aspects in my previous slides. I just wanted to recap that, we started with this and we covered everything. And this slide also we have seen, but this brings us to a different question. So when I presented this slide, I mentioned that the cost of development is around 1 billion to $2 billion. That's around 7,500 to 15,000 crores, which is a huge, huge amount. And the time taken is around 10 to 15 years, and success rate is very low. So how much money the companies are spending on research? Look at these numbers. These are no Indian company will feature here. These are all global top companies, Pfizer being the largest company, Roche, Novartis, these are all big companies. And just look at this budget. This is last year's R&D budget, research budget for Roche. If you see, $9.8 billion. So consider approximately 75,000 crores. That is the amount that Roche, just one company, invested in 1 year on research. This is a huge, huge amount. And just to give you a little context, if you look at Indian industry, the largest Indian pharmaceutical company is Sun Pharma, and the turnover of Sun Pharma is $4 billion. And if you imagine the R&D budget of Roche, I'm not talking about the turnover, I'm talking about the R&D budget. It's around $10 billion, $9.8 billion. Look at the difference. Look at the amount of money that is getting invested. So this brings us to a basic question. Why invest so much money and time in the research? You need to spend 10, 15 years, you need to spend 75,000 crores, you need to, and still there is no guarantee that you will be successful. Why do this research? It's all about risk and reward. And this is true in every walk of life, whether it is research or any part of our life. Risk and reward go proportionately. Higher the risk, higher is the reward. So we looked at the R&D budgets here. Okay, how much Roche is investing is around $10 billion for entire research in a year. Look at the sale of the top 10 selling pharma drugs, last year's drugs, global sale. There is a drug called Humira. It's a monoclonal antibody. Annual sale is around $20 billion, $19.7 billion. Okay. So, example, I just want to give you a little bit idea that the company is investing $75,000 or $10 billion entire research. Just one product is giving them double the amount in 1 year, $20 billion. And imagine this drug gets a patent life of around 15 years. So, since you file a patent 15 to 17 years, you will be the only player in the market. Every year you will make this much money. This is the kind of reward that you can get. So, very resource intensive, very high risk, very high investment, but the returns are very, very good, incredibly good. So, most of the Indian companies are somehow not doing that much of innovative research because the financial pockets are not so deep. But yeah, there are still few companies like Denmark who are aggressively pursuing this research. And with this, I think I complete my talk on the entire process of drug discovery. And I'm also trying to give you a context of why we should really invest in the innovative research. And if any of you students are thinking of making a career in drug discovery, you should know what drug discovery is, and you should know what kind of returns are there in drug discovery, why this area is so hot. I have completed my talk, but because this talk is for the students, I have kept one slide. I want to give a little bit brief information, and then I will open this topic for discussion. So, if any of the students want to make a career in drug discovery and development, what is it that you need? Minimum qualification is masters. If anyone is thinking that I have done my B Farm and I want to get into this research, please do not do that. First of all, you will most likely you will not get an opportunity. Even if you get, you will not grow in your career. If you want to be in this research, minimum masters. In fact, MPharm is not good. You need to do PhD and postdoctorates from US and Europe. They are most prepared people actually for this kind of research. If you want to go for a MBA after B Pharmacy, that's fine. You can still have a role in drug discovery. So B Pharm MBAs can have an important role in project management. You have seen the project last for 10 years. It involves so many different teams. So you need a center point contact, controlling mechanism, controlling team. That's project management or the portfolio management, operations management. So you will not have a technical role, but you will coordinate all the activities. So there is a role for B Pharm MBA as well in drug discovery. And specialization wise, in general, you must have noticed already the medicinal chemistry has got important role in drug discovery, pharmacology, and pharmaceutics. Pharmaceutics people are mostly used in pharmacokinetics, DMPK. So these three functions, specializations are very, very relevant for drug discovery. But having said this, there are a lot of other people who can also work because drug discovery also has like quality assurance, intellectual property, pharmaceutical analysis, cellular and molecular biology, regulatory sciences, clinical, and there are a lot more. But all I'm saying is there is a role for every specialization in drug discovery if you really are keen on joining drug discovery and making it as your career. But I also want to give you a word of caution and give you a real picture. Drug discovery is a very, very hot area in USA and Europe particularly because all the bigger pharma companies are there and their pockets are very deep. They have a lot of financial backing. They do that kind of research. In India, unfortunately, there are very few companies, you can count on fingers, who are engaged in drug discovery. Glenmark being the front runner has been one of the most successful company, and I'm proud to be associated with Glenmark. That we have done a lot of innovative research. We have got an R&D center in Switzerland as well as United States and Mumbai. So very few pharma companies are doing this drug discovery and development. There are a lot of CROs, contract research organizations. Like pharma companies are doing research for their own. The CROs provide the service and they charge you in terms against that service. There are CROs, and there are a lot of multinational companies like Bristol Myers Squibb and Amgen. They have set up their own R&D centers in Bangalore. But on some of them have started to have a collaboration with some CROs in India. So I would say Indian scenario, Indian industry is not so much engaged in drug discovery as of now, but future is bright. Down the line, more and more multinational companies will set up their R&D centers in India because India has got a fantastic talent in terms of pharmaceutical sciences, and the cost of research is very low in India because the manpower cost is very, very low. So the future is bright. But as on date, I think if someone really wants to make a career in drug discovery, Based on my personal experience, I would really advise that you plan for going abroad for studies, do PhD, do postdoctorate research in US and Europe, and it will be a fantastic career for you. So I think that is all I have for you, and thank you so much for patiently listening to me. And I have tried to give you a very superficial, high-level overview of drug discovery and development, and I'm open to take any questions that you have. Thank you. Over to you. Hello.
Hello, good afternoon, sir.
Yeah. Yeah.
Sir, till now we have not received any question. Let me ask again.
Please.
Just a minute, sir. So might be the topic which you have discussed is discussed in very simple manner that they have understand everything, and that's
Yeah.
why we are not doing
I
any
hope that is
question.
I hope that is the case. Sorry, I hope that is the case, and I hope it's not the other way around that they didn't understand anything, so there are no questions. So I
No,
would be happy if they understood everything. That's good.
yes, sir. Yes, sir. They have understood everything. Thank you, sir. Thank you
No
for
problem.
giving us time from your busy schedule. Sir, you have explained drug discovery and development in very simple manner, right from A to Z. That is from
Uh,
target identification to human trial and marketing a drug. Then you have also
right,
explained the various steps in drug discovery, like drug discovery and screening, ADME, target identification, then clinical trial and approval by FDA. Thank you, sir, for
right,
sharing and guiding our student on the careers in drug discovery and development. I assure you that our student will grab the
right.
opportunity in career, and in future, if we if we require any help about this career, definitely we will ask you, sir. Thank you, sir.
Thank you. I will.
So on
I'll
behalf
be.
of
I'll be happy. Sure, I'll be very happy
yes,
if I can be of any help. Even if no one has any questions right now, if there are some questions that come to mind little later, I'm happy to receive them on email. I have absolutely no problem. And I tried to keep this presentation very simple because I wanted to consider students and give them a bigger perspective rather than talking very complex scenarios and making it difficult for them to understand because it is virtual. I even try to reduce the text on my slides and keep maximum pictures so that students can
yes,
understand it
yes,
easily.
yes,
Yeah,
yes, sir. The funnel is actually very attractive point that they can understand right from the discovery till the market, how the procedure in how they are
sure.
requiring. Sir, actually, we don't want any question, but we got feedback from the students, so I want to share it. Just a minute, sir.
Please.
Sir, from Mayuri, she is saying, sir, very helpful session. They understand each and everything. And from Saurav also, he is saying that thank you, sir. That means
No problem.
just yes, that means they have understand it correctly, sir. It the feedback which we
Yeah,
have received. Okay,
yeah. I'll
according
be
to
very
that.
happy if they really understood. Yeah, thank you.
Yes, sir. So you can see this comments that most of the students are saying that they're very easy to understand.
Okay, great. Happy to know
Okay.
that.
Yes, sir. Yes, sir. So though we don't have any questions, but yes, the feedback saying that it's very good session. Thank
All
you,
right.
sir. Thank you for giving us a time. I, Dhanashri Mai, on behalf of Gokul Education Society, I thank you very much. Thank you, sir.
Thank you. It was my pleasure having you again on this talk and happy to help always anytime I can be reached. Thank you so much.
Sir, is it okay if we are sharing your mail ID, official mail ID? If
Absolutely, there
any
is no
student
problem. There's no
okay.
problem. Absolutely no problem. You can share.
Because actually students are asking about the email ID.
Absolutely no problem. Anyone wants to share their CVs, any guidance they want, any help they want, I can assure whatever I can, I will definitely do. No problem. You can share
Okay,
my email
okay,
ID.
sir. I will share this email ID on the group. Thank you. Thank you
No
very
problem.
much, sir. Okay.
All right. Thank you, and all
Okay,
the
sir.
best to all the students for their future.
Thank you, sir.
All right.